Okay, yesterday I talked, there are many methods. Basically, there are 6 methods. 5 methods are very important. That is one part I have discussed yesterday, that is base excision repair. Now, the remaining part is one is nucleotide excision repair. Basically, when the nucleus, this DNA is damaged, a considerable part, there are 2 basic pathways. One is global genomic nucleotide excision repair, and another is transcription coupled nucleotide excision repair. These 2 procedures, you can imagine the degree and number of proteins which are involved in detecting the damage. So, which pathway will be followed will be determined by the initial recognition step of DNA damage. So, Xeroderma Pigmentosum, what I have told yesterday, it belongs to global genomic NER defect. There are other diseases like transcription coupled. If you cannot remember, at least Xeroderma Pigmentosum, write down under the headline of nucleotide defects in the nucleotide excision repair. I have given a list. Lists are huge, but only a few selected one I have given. So, this is a favorite question. At least you tell this nucleotide excision repair defect. So, what is global genomic recognition? This nucleotide excision repair is basically a global surveillance system. Global means not world, the whole genome. Whole genome is basically scanned, and whether they want to see the proteins, want to see whether this pathway will be required or transcription coupled, better I should say the transcription coupled NER is basically required of the areas which block RNA polymerase to transcribe. So, since this is transcription coupled, so it is related to RNA polymerase areas which are transcribed. But global genomic area, it makes a global surveillance scanning the whole genome. It's a genome-wide surveillance system, and it acts as a broad spectrum of repair mechanism. The basic, because when we are exposed to UV rays, but there is a little bit minute, small damages, these can be managed in a different way. Maybe one nucleotide excision repair will be sufficient, but when there is large number of large adducts of DNA defects are there, then this global genomic area comes into the play, and there is a continuous surveillance system because the genome has to be protected from any kind of insult, and it must maintain its integrity for its own function. Second is the basic purpose remaining the same, that you have to excise the damaged segment, then that part needs to be synthesized and that is to be joined. So, both share the same common pathway. In the next slide you will see, only part is the initial recognition step to determine. So, nucleotide excision repair, you see there are 2 pathways. If damage is here, one is transcription coupled, those areas which are transcribed, and other areas are global excision repair. You see the common pathways, these last pathways are almost same common. So, bulky DNA damage which are caused by UV radiation are done by a 3-step method that I told you. Recognition of the defect, and here you see the removal of 12 to 24 nucleotide sequence upstream and downstream. That means a huge area of DNA segment needs to be cut, and then the cutting will be done by endonuclease. Endonuclease is basically a phosphodiesterase enzyme. what does it do? All the nucleotides are attached to each other with by a phosphodiester bond, and that is done by DNA ligand. Now, phosphodiesterase enzyme, basically you have heard, because during the cleavage of ATP or ADP, where the terminal phosphates have been cleaved off, but here again, this endonuclease, basically the endonuclease belongs to phosphodiesterase. This is one subset of phosphodiesterase enzyme you can see, because that bond tends to be cleaved, not that in individual basis or the nucleosides are cut, but the nucleotides are cut, which is joined together by phosphodiesterase enzyme. And then the synthesis of the removed DNA, and this mechanism is evolutionarily conserved because of its necessity. The cell needs to be kept in its original function as much as possible with as much as a normal function. So in prokaryotes, the NER is mediated by UVL protein. It's a large complex of protein, and the term which you might be reading in your books includes exonuclease, not exonuclease, but exonuclease. It's basically a UVR ABC endonuclease multi-enzyme complex. It's endonuclease, but that entire complex is named as exonuclease. But in eukaryotes, much more proteins are involved. So in the books you will read the reference, maybe they are this time the prokaryotic type of this lesion for the nucleotide excision repair, not the eukaryote, because these are large number of proteins are involved. What are the Regina? Regina, have to fast move, go Regina. Mobile. So this is the picture of Xeroderma Pigmentosum. As I told you, in your professional career, you will definitely come across many patients. Only thing is that you have to be a little bit suspicious. So these Xeroderma Pigmentosum, there is pigmentation when they are exposed to sun, and the sun exposed areas are basically damaged. So they are having dry skin. So that is the reason why its name is Xeroderma. And there is pigmentation of the skin also. In addition, since this is a defect of the excision repair, nucleotide excision repair, of course this system is present in all the cells. So there will be some neurological dysfunction, and normally cancer doesn't occur in childhood, it occurs in old age because of accumulation of many mutations. So if they are left untreated, they might develop skin cancer by the age of 10 years. So mismatch repair, this you try to remember a little bit in detail, because that may be asked in during examination. So it is also because if there is an erroneous insertion or deletion or incorporation of bases, and it occurs during DNA replication, recombination, repair and damage. There are many steps where this mismatch repair means a mismatched nucleotide has been placed, and that needs to be detected. So this recognition is done by the prokaryotes by three different enzymes called MUT, MUL and MUH. Corresponding proteins are only two, MSH and MLH in eukaryotes. You see in this slide, in this part, in eukaryotes, the protein involved is mute class proteins. Mutes means mutant type of proteins. So they belong to this three class. So in most eukaryotes, this MUS is corresponding to MSH, and the analog of MUTL is MAH in eukaryotes. Humans are also having a specific type that is called HMUTL and HMLH1. These enzymes are defective in human diseases. So again, this recognition is done by this protein, and this system present essentially in all cells to correct, because replication, this translation, all these processes are going on almost all nucleated cells. And it is consisting of two proteins, one detects mismatch and one rectifies. This is the purpose of having two. You see there are two two proteins. So this is followed again by the same procedure, that removal and synthesis and joining by deacyling by DNA ligase. Pata. So this step, you see, this is the defect. The defect may be at the 5' end, is maybe at the 3' end. Now GATC sequence is required. Now in the normal, this parent DNA, from where this DNA is copied during replication, it is methylated, and that is the catch point of this repair by this method. So there is a protein, this defect is here. So mut protein basically binds to this methylated site, and then it starts scanning. It comes here. Then other proteins come into the play, mut H, and all these proteins come to this defect site, and finally it scans the entire DNA. You see, this part has been recognized, this part has been recognized. Now it is starting scanning. It's scanning this is moving from this part to this part until this CAGT sequence, or here this CTAG sequence is reached. You see, this is moving, and now it is getting fixed. Okay, this is the next step. You see here, the nick is met now here. Defect is here. Nick is made at the 3' end. So a whole lot of this DNA sequence is clipped off. The entire region is clipped off from this side to the region where the defect is there, and then finally it is sealed by the DNA ligase. So if the end, now look at the problem, it is present at the 3' end. So flap is from this, it is scanning from this side, and it is removing from 3' to 5' exonuclease. Okay. In contrast, now you see the mismatch repair here. It was originally defect, I told you. It was here, defect is here. So this DNA helicase enzyme has to unwind, otherwise it will not be able to remove. So DNA helicase will come into the play, and SSB, that is single strand binding protein, will be acting, and the exonuclease, because this part has been removed from the main part by unwinding procedure through a nick, and that causes exonuclease, and the DNA will be synthesized. But remember, synthesis always occurs from 5' to 3' direction. Here, the nick, you see, the problem is here. You see, if the problem was here, now the problem is at the 3' end, and this is at the 5' end. This is here. The defect, this is what I have shown, is on the left side. Now, this problem is there on the 3' end. It was at the 3' end. Now it is in the growing chain at the 5' end. Okay. So here it is. So when it is here, so again the same procedure. It removes from there, and again there you can see the synthesis. It is in this direction only. So it can be at the 3' end, it can be at the 5' end. In either case, first and foremost thing, that bound, that is double-stranded DNA, needs to be unwound through a helicase enzyme, and there will be cut and the synthesis and blocking. You see, the problem is here, cutting is done here, cutting was done here, cutting is done here. Can you see? So next part of the go. So mismatch repair basically involves the detection of methylated DNA in the parent strand, and then it starts scanning the whole process, this mut S, mut L, and mut H, these are present in the prokaryotic system, and then they detect, they remove that part and finally joins. In human beings, one cancer is caused by the defective gene, that is hereditary nonpolyposis colorectal cancer. It might come as an MCQ, which protein is defective. MLH and MSH, these are the two proteins only. But this is specific to human, so specifically HMLH1 and HMSH2 genes are defective. If you cannot remember, at least tell that this is MLH and MSH gene. That will be sufficient for you to answer to that question. Now you see, normally you cannot detect something, some cancer is not easy to diagnose until and unless the disease is quite advanced. Now again, the chronic cancer is a little bit difficult to diagnose because patient may not present in the acute stage. Maybe patient will be coming with the problems of bleeding or rectum or bleeding with the stool, or there may be loss of weight, or there might be some problem with the bowel movement. Usually there will be constipation. So this is a hereditary nonpolyposis colorectal cancer. Basically, these type of patients suffer from cancer syndrome. There are cancers in multiple organs, not only colon, but endometrium, ovary in case of female, stomach, hepatobiliary system, upper urinary tract, even the brain cancer. So the patient suffers from brain cancer because, as I told you earlier, all these DNA defective repairs are present in all the cells. Those cells which are actively dividing, they are supposed to get more errors. And some cancers are specific for some organs. As you grow older or as you are exposed to newer things, you will understand that why some cancers are common in some organs, why some virus affect some parts of the body, not others. Some organisms infect some part of the body, not some organs of the body, not others. So there is a specificity because again there is a molecular interaction. Okay, better. So double-strand DNA breaks. So it's basically hazardous. Double-strand DNA breaks means that helical structure is totally destroyed. It is very difficult because you will not get a complementary copy from where you will get make the DNA replicase, the DNA polymerase will form a new strand because the entire helix is disturbed. There will be cross-link. By chance, if the double-stranded breaks are also accompanied by cross-linkage, means you will not get either strand for replication. So a large segment of DNA needs to be cut. So there are three mechanisms. So it cannot be done by the procedures what so far I have discussed, that is base excision, nucleotide excision, or mismatch repair. Because a huge area of DNA has been damaged. So there are three basic mechanisms. Only you remember this thing: homologous or non-homologous, homologous recombination or non-homologous end joining, microhomology mediated end joining. Don't try to remember because this why you should not remember. I'll tell you. See, this double-stranded DNA breaks are there. It is highly likely that if the body sensor mechanism senses that this cell cannot be repaired, the cell will undergo cell death, or even if it is repaired, they will undergo severe mutation. So double-stranded DNA breaks usually occurs when there are UV rays and X-rays exposure. So large part of the DNA has been damaged. So this is done by homologous recombination. Means if you get a copy from where you can basically make another copy, almost the identical sequence something like meiosis crossover. During meiosis crossover, homologous chromosomes, they pair together and there is a crossing over. So this is the identical sequence which is maintained in the homologous recombination, because the parent strand must be very much perfect in that nature. So presence of an identical or nearly identical sequence is used as a template. And the enzymatic machinery is the same, just like chromosomal crossover during meiosis and repair using a sister chromatid. It is usually available after the D2 phase during replication or on the homologous as a template. In contrast, non-homologous end joining, the exchange of material occurs randomly, and because of random presence of all those nucleotides, there is a high chance of mutation. These two strands will not be identical. So, especially important, before the cell has replicated for DNA, it relies on, basically it relies on short homologous sequence. Here it is an identical DNA sequence. That is the reason why you will get an identical copy. But here is a short sequence of homologous sequence based on which the recombination starts, and because the randomly it occurs, it can cause mutation during repair, it can have deletions, it must non-matching, and one important enzyme, that is a special type of enzyme, is involved here, that is DNA ligase 4. If you can remember, it is not that DNA ligase which is normally we use in other forms of repair mechanism. So, double-stranded DNA brand, remember the two proteins, one is KU and DNKP protein binds. It binds to the both ends, and then the both ends tries to approximate to each other. So, they try to approximate to each other, and then one gets unknown and the replication starts, and you can see the formation of the new strand. It is followed again by the same ligase enzyme. You read it from the book, it will be clear to you. What are they called? So, microhomology mediated end joining is one part, but why I am not telling? Because it is accompanied by a deletion. So, it is a mutagenic pathway. So, mutagenic pathway is not a very useful method for DNA repair. It can repair, but if it causes mutagenesis, it is a mutagenic pathway, it will form either a tumor or a cancer. So, why the body will adopt such a procedure? But there is one procedure, this one procedure just I have written. It is not, don't try to remember, that's just for your information I have written. Another part, so disorder due to defective strand repair, I told you, this is homologous recombination or non-homologous end joining. Breast cancer is a common problem. So, breast cancer susceptibility, in the chart I have shown, BRCA1 and BRCA2 genes are there, Bloom syndrome and Werner syndrome. So, breast cancer susceptibility genes are because of the homologous recombination. But severe combined immunodeficiency, during immunology class I told you that if both the pathways, that is active immunity, passive immunity, all these things are blocked, it is blocked because there is a gross disturbance in the non-homologous end joining repair. Joining is there, but it may be defective. What to the direct repair? Direct repair usually doesn't occur, although I have described for your own understanding. I am telling you it is present in the bacteria or in the lower prokaryote. It's prokaryotes, not in, no longer function in humans. I am telling you these, because these bacteria or the prokaryotes are also exposed to UV radiation, so they can have an UV radiated induced pyrimidine dimer, and photoreactivation basically directly damages this, causes formation of DNA dimer, and it requires an UV light of the range of 300 to 400, I must say, not 500. Photolase is the enzyme which is responsible for this DNA repair. Photolase is an enzyme present in the bacteria, fungus and most animals, but this enzyme is not present in human being. And the enzyme concerned for DNA repair is methyl methyltransferase. It's an expensive procedure in the sense that every time one MT enzyme will be required. Normally, enzyme is a catalyst, performs catalysis, means the enzyme remains there, it catalyzes the reaction without being destroyed. But in this particular case, every time this MGMT enzyme will be required. So this reaction is basically stoichiometric, just like other chemical reactions, which is dependent on the reactant amount rather than catalytic. Because if it would have been catalytic, then the enzyme would have been there, and only the addition of the reactants would have led to formation of products. So certain methylation or basisations of cytosine and adenine, these are the three main basic steps of direct repair. So direct repair doesn't exist in human beings. So direct thymine diameter repair pathway, I am telling you, the enzyme here it is called DNA photolyse. So it is cleaved basically the pyrimidine dimer. So there is no such direct removal in higher animals, including humans. You remember that thing. So this is for the prokaryotes. So as I told you yesterday, that this type of questions will be there. Now today I am going to the next part, the main part. Hopefully I will be able to cover within half an hour time, because this is cross Now mutation you try to understand, you will understand it is not that difficult. And what I would say, whatever books you are reading, at least try to read this molecular biology or the genetics part from Harper books, and try to memorize the figures. And Harper books, all teachers all have read Harper books. They will remember all those things, and good charts are there. I have checked the book also. So I think this will be informative, particularly the mutation part, and this mutation part is written in a very abridged form within 2 pages. It will take about 15 to 20 minutes for you to read that chapter, and the 3 charts. You just remember 3 figures, that will be sufficient for you. This is what I am going to tell you. In addition, something more than what is written in the book. Not only the latest edition, that is 32nd edition, 31st, 30th edition. If you go, one more important information of the cystic fibrosis is not there in the 32 edition. This I have checked it, but it is present in the other editions. Okay. Mutation. Okay. You see types of DNA damage. I told you yesterday what are the different types of. Try to remember that chart. It will be available in the book. Again, this chart is available in the Harper book. So germline mutation. Yesterday I explained. Again, I am telling you, germline mutation means those mutations which are occurring in the germ cells, germline cells, testis and ovary. That is spermatogenesis, oogenesis. So if there is a mutation in the sperm or ovum, this will be transmitted to the next progeny. So this is all the diseases are inherited diseases, because they are transmitted from the parents through that sperm or the ovum. So not only this hemoglobinopathies or mutation, any kind of diseases, including defective genes which is already present, they are inherited. Somatic mutation occurs after birth. I told you, we are exposed to a hostile array of insulting agents which are, or damaging agents I would say, which are causing different types of damages in our body, including damage to the DNA. And if you go to the radiology department, you will see that particular sign is there. Normally, any woman during the childbearing age are not allowed to undergo exposure or CT scan until and unless it is absolutely indicated. So protect those, particularly those males and protect during the childbearing age. Do not move to the radiology department or do not get exposed until and unless it is required. Of course, the people are working there, but there is a radiation band which is placed onto the chest, so that every month, since I was in charge of radiology department of our hospital also, we used to send that badges to one center which is authorized by the Bhabha Atomic Research Center. They used to monitor it, how much degree of exposure is there, because beyond permissible limit, this is not allowed. So, protection of pregnant women particularly, because fetus is growing at very high speed. So, there is a high chance that the fetus is likely to have a mutation. So, these X-ray, CT scan machine, including radioactive particles, and they cause basically defects in the gonadal, and it's called germline mosaicism, is altogether a different part or genetic issue I can discuss later, but only remember germline mutation and somatic mutation. So, in the body one might have different types of one mutation is there in the body, that is somatic mutation, another mutation is there in the germline. So, basically this is a mosaic type of pattern. So, that is a germline mosaic, because body cells are not defective DNA, only the germline, so that is called germline mosaicism. So, definition, what is mutation? So, any change in the nucleotide sequence in the DNA is a mutation. Now, we have the DNA replication, that DNA polymerase or the eukaryotic, the 5 parts I told you, lagging strand, leading strand, they do have true reading capacity, but there it is, every system is not perfect. So, there is a chance of error. So, the statistical frequency probability is 1 mutation per 10 to the power 6 cell division, means almost 10 lakhs. And mutagens like physical and chemical agents, and they are classified in 3 different types. One is substitution, one is deletion, and another is insertion. Wrongly, one thing can be substituted, nucleotide can be deleted, nucleotide can be inserted as well, because of the faulty procedure. So, point mutation, you try to remember. Point mutation, that means that single base is altered. It can be of 2 types. One is transition, one is called transversion. What is transition? Means change of single base. It can be either purine to purine or pyrimidine to pyrimidine, another pyrimidine. So, we are having A, adenine and guanine, instead of A, if it is G, or instead of G, if it is A, or C and T. If it is a C, instead of C, it is a T, or T instead of C, then that is called transition. In contrast, transversion means changeover from purine to either of the pyrimidines, or changeover of pyrimidine from either of the purines. So, the chance is there, A can have a transition from A to G, C can have a transition between C and T. This is called transition on either side, but transversion, that means A can become T, A can become G, or A can become C also. This is transversion. Similarly, G can become C, G can become T, and in the reverse direction also, G can become C, G can become A, G can become T, G can become C. So, you remember that One is transition, one is transversion. These are the basic questions. If somebody cannot answer anything, then this simple question is just tell me what is transition and what is transversion. So that's it. Next. So one thing I just giving you this chart I have shown you during genetic coding. I am giving a reference for your understanding. See, one codon is there for AUG, there is a single codon, but there are many codons which are basically coding for one particular amino acid, and that is what is called degeneracy. See, there is a mutation going on, but because of the degeneracy, there may not be any effect, because the same nucleotides may be mutated, but since we are having a buffer system, the double codons are there, triple codons are there, 4 codons are there, 6 codons are there. So instead of that nucleotide which is coding for valine, if it is coding for same valine, because there are 4 codons are available, so it will not have any effect. So there may not be any effect. And another part is that is the codon representing more than one codon representing one amino acid is called degeneracy. Remember. Second part, what is that? Second part is oobling. The third nucleotide sequence is not of much importance. So if mutation occurs at the third position, it may not have any effect, and body has basically evolved this mechanism as a buffer system, so that the wrong amino acid is not incorporated finally into the during translation because of the degeneracy, because of the oobl phenomena. So again I am telling you the classification of amino acid. Try to remember importance here, because if the amino acid, if there is a mutation, and if the same amino acid of the one amino acid from the same group is replaced, so functional groups are remaining same. There is no alteration of the functional group. So although the amino acid has been changed due to mutation, functionally that protein will not be that much altered if it belongs to the same class. But if it belongs to the other class, then there will be a problem. And in a short while you will see that thing. So that is the reason I have shown it to you. So because of the mutation, first and foremost we are having a buffer of multiple codons, we are having oobl, and third point is we are having a reserve also, different kinds of amino acid, different numbers of amino acids which is present in the, which can replace. Second. So see, there is no mutation. See, there is a, here I am telling you the 3 points I have taken. One is DNA level, mRNA level. So there is no mutation, lysine is there. This is silent mutation. You see, it has been changed. As I told you, this is the third position. So because of the oobl, of the same codon, this lysine, although there is a mutation here, T2A means it's basically transversal. And here you see the nonsense codon has come. A has become U and a stop codon. UAG is a stop codon. So if it is a stop codon, then of course there will be premature termination of protein synthesis, and it will be a truncated protein of varying length. Missense means you see here PCC is there, AGC here, it is coding for arginine. So it is a missense mutation, and again this is a missense mutation. You see TCG, TGC and ACG is coding for threonine. So if there is a mutation here, you see C is converting to G, cytosine to guanine. So it is causing an arginine, here it is causing threonine. So this is causing a missense. This is not the same thing. But here you see, this is lysine same. Although there is a mutation, the amino acid, that means the outcome remaining the same. What is that? So impact of mutation, it can be silent. There will be no effect, no detectable outcome because of the degeneracy, because of the oval. Change in the third position, these 2 points I have shown in the genetic code, just to remember. So silent mutation, there will be no detectable effect because there will be no change in the amino acid sequence. Translation will occur as usual. Impact of missense mutation, by chance, if a wrong amino acid has been placed because of the mutation, so it can be acceptable because of the buffering action, as I told you, because if it is of the same group, so it will not cause much disturbance because of the presence of the functional groups. Second, it can be partially acceptable, there will be compromised function. It will function, but it will not function normally. So some abnormality will be there. Again, I will give you an example. One is unacceptable, that means that cannot function at all because that is present at the very crucial part of that protein. So it will not allow either the folding or it will not allow the protein to function properly. So that is unacceptable. So if impact of missense mutation are 3, one is acceptable, one is partially acceptable, one is unacceptable. What are they? So again I am telling you, you see the aliphatic amino, this chain I am telling you. So dibasic amino acid is lysine, amino acid with an amide group, that is asparagine. So if since there is a 2 amide group is there, but here it is a single amide group, so basically there may not be any change and it doesn't happen. Acidic amino acid is glutamic acid, one more acidic group is there. But in contrast, branch chain amino acid, valine, is an aliphatic amino acid, is monocarboxylic acid. So only one carboxyl group is there, but here it is one more additional carboxyl group is there. So that is the reason why it is an acidic. So it will cause a disturbance because functional groups are not same. Again, heterocyclic amino acid is histidine. If you recall the structure of hemoglobin, it is having an imidazole ring and which is maintaining that iron in the ferrous state and it is helping in the binding of oxygen. Now, if that histidine is replaced by tyrosine, obviously you will not get that group. So it will not function. I am telling you what is that. Now you see here, acceptable. So as I told you in the previous slide, lysine is at position number 61. There is a mutation. Lysine is replaced by arginine, but it is changing from normal hemoglobin to hemoglobin Hitkari. So there will be normal function, but because of the abnormal amino acid, the electrophoretic mobility will not be the same. A person will be defective, 2%, the white person, I may have some problem somewhere, but functionally I am normal. Suppose my leg has been amputated, but I am now with the crutch I can function, or with some support I can function, artificial leg I can function. So functionally I will not be abnormal. But if you look at the body, basically I am abnormal. So that is what functional part we want to see, and we basically, regardless of the structure, basically we are concerned with the functions of that particular protein as far as the biological activity is concerned. Same thing is true for structural proteins also, because structure has to be maintained. So that structure is required for the functional part of that, maintaining the structure. Say partially acceptable. You see, hemoglobin B, you remember this part, and again I will tell you in the next slide because this is a question. Hemoglobin A, beta chain, and this partially acceptable hemoglobin S, valine. So glutamic acid is replaced by valine. Glutamic acid, I told you, it is an acidic protein. Aliphatic amino acid is a valine. It doesn't have an extra carboxyl group, so it is partially acceptable. Unacceptable is alpha chain, that is hemoglobin Boston. So distal histidine is replaced by tyrosine. So this hemoglobin will not be able to function at all. So there will be death. Baby will die. And that is called hemoglobin M, HbM, Boston. It involves alpha chain. It will not be able to transport iron, the oxygen at all. So if the oxygen cannot be transported, cell will not get supply, and it is basically unacceptable, and there will be death also. Ready? So structural hemoglobin, I told you that you remember this structure, this imidazole ring, it is maintaining the iron in the ferrous state, and it is also carrying oxygen. So histidine is replaced by the tyrosine. So this structure, the imidazole structure, nitrogen is not present in the tyrosine, and that is the reason why it is incompatible with the life, and that is an unacceptable missense mutation. So it can be missense because outcome encoding a different amino acid. So DNA here, glutamic acid is replaced by the valine. HBS binds, hemoglobin S binds to the oxygen, but precipitates when it is deoxygenated. And when it is deoxygenated, it sickles. So this is the basis of sickle cell anemia. So I told you what time. At the sixth position, you remember this slide, this is the, you should come in a question. So genetic defect of the sickle cell anemia, it's an autosomal recessive. Two types are there. One is heterozygous and one is homozygous. Homozygous form is severe because both the chains are having hemoglobin S. So at the sixth position of the beta chain of hemoglobin, this was told, I think, during amino acid class. Glutamic acid is a polar amino acid, is replaced by valine, which is non-polar. So forming the sickling or the sickle cell of the RBC. So HBS cannot carry oxygen, subnormal function, or with low PO2, and sickle RBC. Normally, RBC can mold during passage through the capillaries, but sickle cell will not be able to move through that capillary and the small vessels. So they might get blocked. So there will be a thrombosis of that area. That area will not be distal to that area where the block will be there. So that area will remain underperfused, or that might undergo necrosis. So in high altitude, when there is less oxygen supply, cells are likely to be sickled in those patients who are suffering from sickle cell anemia. Even in the normal situation also, when the oxygen supply is less, when the patient is suffering from some disorder which cannot deliver adequate amount of oxygen due to pulmonary disease or cardiac disease, the patient can also have sickle cell crisis. This crisis means a painful crisis if it occurs in the bone. A huge part of the bone will not get adequate amount of blood, so there will be pain in the bone. It can cause vaso-occlusive crisis, causing infarction. Sequestration crisis, aplastic crisis, stroke, you will see if it occurs in the brain. So there will be microinfarct. You will not be knowing the patient might be having some severe cerebrovascular accident, sudden unconsciousness, because that sickle cell has blocked the microvessels or the capillaries of the brain. So premature death of the RBC because of the short span of, like all hemoglobinopathies, if it is a little bit severe, depending on the degree of severity, there will be varying degree of lifespan. Some heterozygous trait will having a longer RBC survival because one gene is normal, one gene is sickle. But in contrast, sickle cell having both the genes are sickle, so both the strands will be of S type, so their survival is less. একটা ভিডিও আছে পরে দেখাচ্ছি, পরেটা দেখাচ্ছি। So global distribution of hemoglobinopathy is a common disorder you will see, and you will be seeing in the clinical cases also. This can cause defects in the, if it codes for globin gene, that is called hemoglobinopathy, that is thalassemia. পরেটা দেখা। So normal hemoglobin is 2 alpha 2 beta alpha chain abnormality. If it is absent, then this is incompatible and there will be intrauterine death. So production of non-alpha chain varies with the age, fetal hemoglobin. This non-alpha chain means two beta chains are fixed. Alpha chain during fetal life we are having a HbF, having gamma chains. So disorder affected the beta chains do not present until the 6th month of age. So hemoglobin A2 is another form which you will also see, that is called having delta chain. You try to remember, and that is present usually normally less than 2.5 to 2%. It is made from constantly from birth, but this amount is very less. So qualitative abnormality, if you see, when I was an MD student, it was the number was written around 200, 250. When after 10 years, the number was 300, 350. In many books, this was written. Now I am seeing the number has been increased to more than 1,000 types of different types of abnormal hemoglobin. But all these do not cause disease, because as you have seen, there will be no effect at all. So although they are electrophoretically abnormal, structurally abnormal, but functionally they may not cause any disease. So there were some variants which cause diseases like HBS, HBC, HBD, and hemoglobin D, Punjab. So these are detected by electrophoresis, these are detected by HPLC, there is a separate method. So quantitative abnormalities, if there is a amount, so that is called a thalassemia. So alteration of the ratio of the non-alpha chain, in alpha thalassemia, excess beta chain is present, that is incompatible. Beta thalassemia, excess of alpha chains are present. Thalassemia patient you will see, thalassemia major, thalassemia minor, thalassemia intermediate, even carrier also. That heterozygous is called carrier also. So three types. The excess chain precipitates. Almost coming to the end. So nonsense mutation means there is appearance of the stop codon somewhere. So it will be a premature termination of codon. CFTR gene, you try to understand that there is a deletion called F50A deletion. You try to remember only this part, that 5 classes of CFTR gene is there. CFTR is concerned with the transport of chloride, and because of the mutation, there will be an alteration in the CFTR gene, and this will cause a deletion effect, and there will be basically, because of that thing, phenylalanine deletion, phenylalanine will be positioned at 508. So 3D shape of that CFTR gene will not be maintained. So to get a 3D structure, as I am telling you, I told you, it is in a critical position. So this cell will not, this protein will not function. This is a clear example of CFTR gene because of a single amino acid substitution at a very important location. The cell will not be able to assume the 3D structure, and it will not be able to function properly. So this disease is not common here, but it is common in the USA. So frame shift mutation occurs if there is by chance there is an insertion. So if there is a frame shift mutation means if there is a mutation here, either insertion, deletion. So reading frame is from 3 prime, 5 prime end to 3 prime end and non-punctuated. If there is a deletion of particular protein, particular nucleotide, the reading frame will be altered. Because of the alteration of the reading frame, the amino acid sequence distal to that part will be garbled. So one example I am telling you, you see the normal structure is here, the C is removed here, so phenylalanine, proline, threonine, phenylalanine, glutamine and proline. So one amino acid is out. So insertion of a single codon, that is appearance of a stop codon. It can, this is a normal sequence, the stop codon is here. Now because of the insertion of one codon, one nucleotide, you can have a premature stop codon, and the rest of the area is garbled. Because reading frame is altered, that is called frame shift. The name has come, frame shift means it is altering the reading frame of the nucleotide sequence. So second part So একটা যদি 3 prime end এখানে insertion হয়ে যায় -3, এই দেখো এখানে একটা insertion হয়েছে, so stop codon is here, stop codon has come here, stop codon may not come, stop codon may come at a distant place also. So again if it occurs around this stop codon, transmission signals are there to stop protein synthesis, so premature synthesis, stoppage of protein synthesis may also occur. What are they? So some facts related. So the basically, if there is insertion deletion, this will alter the reading frame, and the reading frame will remain so until and unless that is correct, rectified. So it might lead to a stop codon prematurely. It can even exceed the stop codon. So an altered protein will be formed because of the frame shift mutation. By chance, if one codon has been inserted, and after some time one codon has been deleted, so in between part will be garbled. Similarly, two codons insertion, two codon deletion subsequently, intermediate part will be garbled. If one codon, that means 3 triplet sequence is inserted or deleted, and then again reinserted, again that intervening portion will be garbled, not the distal one. All these things will lead to an abnormal protein, and if the number of such insertion deletions occur, and that will cause the epithelial disease, because it will not be normal protein. Okay. Yesterday I showed you the question. First question was on DNA excision repair. Second question was classify mutation. Explain the consequence of point mutation with a suitable example. The example is here, sickle cell anemia. The best example which you can write. So classify mutation. So this is the basic part I told you. Okay.